Hello, my name is Rory Boyle. I'm a postdoctoral researcher in the Penn Frontotemporal Degeneration Centre in the University of Pennsylvania, and I'm going to give a quick overview of our project funded by the Penn AI Tech, which is titled Understanding Aging and Alzheimer's Disease and Related Dementia Disparities using a Representative Epigenetic Clock. And today I'm presenting this on behalf of our team, which consists of Hao Zhu, Wanding Zhu, Anurag Verma, and Corey McMillan, and we're all based at the University of Pennsylvania and/or the Children's Hospital of Philadelphia. So our project is motivated by the consistent findings and well-documented findings of racial disparities in aging and Alzheimer's disease and related dementias, where Black adults in cohort studies are found to have a higher risk of dementia and also a higher risk of physical frailty compared to white adults. And the weathering hypothesis provides an explanation for these disparities. It holds that they arise from the cumulative negative impact of repeated exposures to socioeconomic adversity, discrimination, and marginalization. And with a tool called epigenetic clocks, we can evaluate this hypothesis and investigate the biological mechanisms of these disparities. So very briefly, epigenetic clocks use DNA methylation data to estimate biological aging. DNA methylation is a process where a methyl group is added or removed from the DNA sequence, and this process tends to consistently change over time. So, capitalizing on this, we can use machine learning to predict age based on these methylation profiles. And with our biological age predictions, we can compare them to chronological age to quantify how much older or younger a person appears based on their DNA methylation profile. And in line with the weathering hypothesis and data from two separate cohorts, the Penn Aging Brain cohort and the Penn Medicine Biobank, we've observed that Black older adults in the US show accelerated epigenetic aging compared to white older adults. And this replicates findings in earlier work from our lab in the US Health and Retirement Study. However, the epigenetic clocks that we have used to date have been trained in cohorts that are almost completely comprised of white adults of European ancestry, so the data sets have not been representative of non-white adults. This is an issue because machine learning models don't usually perform well when applied to data that was not adequately represented in the training set. And in the case of genetic modeling and epigenetic modeling, this is an issue as most of these white adults are of European ancestry, and it means that genetic predictors or predictive models may not perform as accurately when applied to individuals with less similar genetic ancestry. In the case of DNA methylation, there is evidence that DNA methylation levels can vary by genetic ancestry, and we know from other types of genetic predictive models like polygenic risk scores that the accuracy of these scores can decrease in line with the genetic distance of a person from the average genetic ancestry of the training set that was used to develop that score. And this is highlighted here in the plot from a paper in Nature by Ding and colleagues in 2023. So with this in mind, it's unclear if the black and white disparities that we've observed with epigenetic clocks reflect true differences in biological aging or simply reflect poor model calibration to underrepresented individuals in the training set and a failure to account for genetic distance. So to resolve this issue, our first aim is to build a representative genetic distance adjusted epigenetic clock, which we will call the REPI clock. And we will validate this in the Penn Medicine Biobank. And this aim will allow us to identify the most appropriate epigenetic clock for application to diverse datasets such as the Penn Medicine Biobank. And we'll use this clock in the Penn Medicine Biobank to better understand ADRD and aging disparities. So there's some initial evidence that epigenetic aging relates to plasma biomarkers of ADRD as well as cognitive impairment, as we have found in preliminary data from the Penn FTD Centre. And there's also evidence from other studies that accelerated epigenetic aging is related to physical frailty, so other aging outcomes. But we don't really know what these associations are like in real world data, nor how generalizable they are to more representative and diverse populations. So to this end, we'll apply the REPI clock to DNA methylation data from the Penn Medicine Biobank, which is a diverse electronic health record linked data set. And we will use this to assess whether epigenetic aging relates to real-world ADRD outcomes and aging outcomes. And our proposal leverages key technological innovations to achieve these aims. Firstly, we'll use software developed by our team member, Wanding Zhu, to process and analyze the data using the Sesame package as well as the M liftover package to harmonize the DNA methylation data from different cohorts across different arrays. And we'll measure or we'll obtain measures of DNA methylation in the Penn Medicine Biobank using the state-of-the-art new Illumina DNA methylation screening array. And again, one thing led to development and validation of this array, which allows for ultra high sample throughput at lower cost per sample while screening for more traits per probe site. In terms of modeling, we'll use a cross-validated elastic net regression to predict age and develop our epigenetic clock model. The elastic net is particularly well suited for high-dimensional data and has been found to outperform deep learning methods for age prediction from blood-based DNA methylation profiles. To quantify genetic distance, we use a data-driven principal components analysis. This is a biologically realistic approach because it will respect the fact that humans will exist along a genetic ancestry continuum and will provide a continuous measure of genetic distance. To characterize our phenotypes, our team member Anurag Verma, who is an expert in the use of electronic health records for phenotype characterization, will support the characterization of cognitive impairment and aging phenotypes in the Penn Medicine Biobank based on the use of ICD codes. In the Penn Medicine Biobank, we'll also obtain measures of ADRD pathology using scalable and less invasive plasma biomarkers. So our finished product will hopefully be a representative and validated Repi clock, and we'll make our model parameters based on available data freely available to others to use on Zenodo.com. And to accompany this, we'll make an open source GitHub toolbox for validating and applying this model. And to increase the usability of these toolboxes, we'll create a guide or video tutorial to accompany the code, and we'll make this freely available on YouTube. So this is stuff that I've done previously with neuroimaging predictive models, and again, we'll apply the same principles here to this project. In terms of the data that we'll use, our representative multi-cohort training set will consist of data from the Health and Retirement Study, the Atherosclerosis Risk and Community Study, and the Women's Health Initiative. Our diverse validation data set will consist of data from the multi-ethnic study on atherosclerosis, and we'll use middle-aged to older-age healthy adults from these cohorts to build our epigenetic clock. We have standing data access approvals in place for all, but one of these cohorts to date, and our data access is pending. We're now waiting to obtain these data. So this is a secondary analysis of existing fully de-identified data, and we'll use this to develop our open source model and apply this our clock in the Penn Medicine Biobank dataset, which is a clinical cohort, and we have access to this data in place with an existing IRB protocol. So thanks very much for taking the time to watch this brief overview of our project. Please do get in touch through the Penn AI Tech team if you have any questions or would like to get updates on this project as we progress through. Thank you very much. Bye bye.